In a sealed vault in Texas sits a handful of black dust scraped from an asteroid that drifted through space before Earth existed. Scientists have now confirmed something remarkable inside it. Every molecular letter used to write DNA and RNA, sealed inside a rock that never touched a living thing. A second asteroid, gathered by a different country's spacecraft, holds that same alphabet. Neither rock contains life. Both contain its ingredients. For over a century, one idea has argued that life's chemistry did not begin on Earth at all, that it was assembling itself in the cold dark between the stars before our planet had an ocean to hold it. Tonight, we trace that idea from a disputed guess to a locked drawer holding pieces of the early solar system. If stories like this one help you drift off, or help you see the universe a little differently, liking this video and subscribing costs you nothing and helps this channel keep finding stories worth telling. Get comfortable now and let your eyes rest. In 3, 2, 1, liftoff. Picture the Earth as it was in its infancy, long before any living thing ever looked back up at the sky above it. The oceans were only beginning to gather, pooling into shallow, restless basins beneath a sky that held none of the oxygen we breathe so easily today. Much of that early water likely arrived from beyond the planet itself, delivered over millions of years by icy, water-rich bodies falling out of the sky, the same general family of objects. We will spend the rest of this story getting to know far better. The air hung heavy with carbon dioxide and nitrogen, laced with the sulfurous breath of volcanoes that almost never seemed to rest, and the young sun itself, dimmer then than it is now, gave this restless world less warmth than it gives us today. One of several reasons scientists still find early Earth such a difficult place to picture clearly. Meteors fell far more often than rain falls now, some burning away as harmless streaks of light, others large enough to reshape entire stretches of the young crust in a single blinding instant. Not so long before this, in cosmic terms, a body roughly the size of Mars is thought to have struck the young Earth, a glancing blow so violent that the debris flung into orbit eventually gathered itself into the moon. We still watch rise over the horizon most every night. This was a newborn world, barely finished forming, still warm in places from the sheer violence of its own birth. Geologists have a name for this earliest chapter of the planet's history, the Hadean Aeon, borrowed from Hades, the underworld of Greek mythology, because, for a very long time, scientists simply assumed conditions here had been too hostile, too infernal, for anything at all to have survived them. For what may have been several 100 million years after that, many scientists believe the inner solar system remained a dangerous, unsettled place, a period some researchers describe as one long, drawn-out demolition, when leftover debris from the solar system's formation kept slamming into every world within reach, Earth very much included. The moon still carries the pockmarked scars of that violence plainly on its face, since nothing on its dry, airless surface ever wears those scars away. Earth's own scars from that same era have long since been erased by rain, by wind, by the slow churn of its restless crust. But there is little reason to think our planet was somehow spared while its smaller neighbor absorbed every blow alone. Only once that bombardment eased, sometime in the neighborhood of 3,800,000,000 years ago, could Earth's surface have offered anything resembling a stable, sheltered place for chemistry to settle down and do something interesting. Scientists know as much as they do about this violent period, largely thanks to another chapter of American ingenuity, the astronauts of the Apollo program, who carried home several hundred pounds of lunar rock and soil during their visits to the Moon, samples that laboratories on Earth could finally date directly rather than merely estimate from a distance. Many of those samples, formed or reset by the heat of ancient impacts, cluster suspiciously around that same narrow stretch of time, which is a large part of why so many scientists came to suspect that both the Moon and the Earth endured a shared, intense pounding right around that period. And yet the oldest convincing traces of life that scientists have found in the rock record date to roughly 3,500,000,000 years ago, preserved in layered mineral formations in Western Australia, known as stromatolites, built, quite literally, by colonies of ancient microorganisms that trapped fine sediment layer upon patient layer, the way a coral reef slowly builds itself today, confirming that formations like these truly were built by living things rather than shaped by some purely mineral process that only mimics biology. Took scientists a great deal of careful, patient work. They looked for the particular wavy, dome-shaped layering that only tends to form when a living mat of microorganisms traps sediment a little at a time, and they searched for the telltale chemical residues that this kind of ancient biology tends to leave behind, signatures that a lifeless mineral process could not easily reproduce on its own. It is a theme that will return throughout this story, telling the genuine article apart from something that merely resembles it, using nothing but careful, patient measurement. Remarkably, living stromatolites still grow in a few shallow, salty bays around the world even now, offering scientists something rare, a living, breathing. Look at what may be one of the oldest surviving arrangements life ever settled into. The best-known living examples grow today in the shallow, unusually salty waters of Shark Bay on the western coast of Australia, where the water is too salty for the small grazing creatures that would otherwise scrape the mats down to nothing, allowing the stromatolites there to keep building, layer by layer, largely undisturbed, much as their ancient ancestors once did. Some claim traces found in far older rock formations in Greenland would push the origin of life back further still, though those particular findings remain genuinely disputed among geologists, and a fair telling of this story has to say so plainly, rather than pretend the matter is settled simply because an older date makes for a more dramatic opening. Even using the more cautious, widely accepted figure, though, the arithmetic left behind is an uncomfortable one. That leaves what may have been only a few 100,000,000 years, a strikingly modest window by the standards of a 4.5-billion-year-old planet, for ordinary chemistry to organize itself into something recognizable as biology. To understand why that timing troubles so many scientists, it helps to appreciate just how much needed to happen inside that window. Even the simplest living cell known today is a marvel of coordinated machinery, a boundary that lets some things in and keeps others out, a way of storing instructions, and a way of reading those instructions to build the very machinery that keeps the whole arrangement running. Scientists studying this problem have long puzzled over a particular chicken and egg riddle buried inside it. The molecules that store life's instructions today are not especially good at doing anything with them, while the molecules that do the actual work are not especially good at storing information for later. One long-standing idea, often called the RNA world hypothesis, proposes that an early molecule capable of doing a little of both jobs at once may have bridged that gap. But even that idea only explains part of the puzzle. Even the order of operations troubled researchers. Proteins, the workhorse molecules that do most of a cell's actual labor, are built following instructions carried by genetic material, yet building that genetic material in the first place typically requires proteins already there to do the building. A bit like needing a fully staffed printing press to produce the very first blueprint for how to build a printing press. None of it assembles itself in a single lucky afternoon, and each piece tends to depend on other pieces already being in place, which is part of why so many researchers have quietly suspected that a few 100 million years, however long that sounds to us, may simply not have been enough time for every piece of that machinery to have been invented from nothing, entirely within Earth's own restless, bombarded, newly cooling oceans. The traditional answer to where life came from has always looked downward, into Earth's own early waters, rather than upward toward the sky. Charles Darwin himself, in a private letter written in 1871 to his friend and fellow naturalist, Joseph Hooker, mused that life might have first stirred into being in what he called a warm little pond, some shallow puddle rich with the right salts, the right gases, and enough gentle energy, perhaps sunlight, perhaps a passing bolt of lightning, to coax the first simple building blocks of life into existence out of nothing more than ordinary chemistry. Even Darwin hedged his own idea carefully, noting in that same letter that nothing of the kind could happen on the Earth of his own day, since any such delicate, newly formed compound would be devoured or absorbed almost instantly by the microscopic life that already blankets our planet now. It was a modest, almost cozy image for something so enormous, and it planted a seed that would occupy scientists for the century that followed. That seed bore its most famous fruit in 1952, when two chemists at the University of Chicago, a graduate student named Stanley Miller and his supervisor, Harold Urey, built a sealed glass apparatus meant to recreate what many scientists at the time believed had been Earth's early atmosphere. Water boiled in one flask to represent the young ocean, its vapor rising into a chamber filled with the simple gases they suspected the early atmosphere once held, while electrodes above sparked continuously, standing in for the lightning that would have lit up those ancient, cloud-choked skies before the whole mixture cooled and condensed and dripped back down to begin the cycle again. When they finally opened the apparatus after days of this and examined the reddish residue that had collected inside, they found something remarkable waiting for them. Amino acids, the same humble molecules that link together in longer chains to build every protein in every living thing on this planet, from the simplest microbe to a whale. For a time, that single laboratory result felt like the final word on the subject. It seemed to prove that Earth's own chemistry, left alone in the right conditions and given enough patience, could climb the first rungs of life's ladder entirely unassisted, with no help required from anywhere beyond its own atmosphere. Even so, the Miller and Urey experiment, for all its fame, only ever solved one part of a much larger puzzle. Amino acids are essential, but they are not the whole story. Life also depends on a separate family of molecules, the nitrogen-rich compounds that spell out the genetic instructions inside RNA and DNA, along with the simple sugars that help hold those instructions together in a workable structure. Decades of follow-up experiments, using every reasonable variation on Miller and Urey's original recipe that chemists could think to try, struggled to produce those particular molecules with anything like the same ease. It was one thing to explain where the building blocks of proteins might have come from. It was quite another to explain where the building blocks of the genetic code itself, the actual letters of life's instruction manual, could have arisen from Earth's own chemistry alone. And the deeper scientists looked into Earth's actual early chemistry over the decades that followed, the more they came to appreciate how narrow, how specific, and how easily disrupted those first true steps toward life would have needed to be out in the real world. It is one thing to produce a handful of these molecules inside a sealed glass vessel built specifically to favor their formation. It is quite another thing entirely to explain how those same molecules, scattered and diluted across an entire early ocean, exposed to storms and tides and the endless churn of a restless young planet, ever found one another in sufficient concentration, assembled themselves into more complex structures, and eventually stumbled onto the trick of copying themselves, all within a window of time that may have been shorter than many scientists are entirely comfortable with. And so, quietly, at the edges of respectable science, a different sort of question began to take shape, one that looked in the opposite direction from Darwin's pond. What if some of the raw ingredients life needed did not have to be invented here from nothing at all? What if at least part of that molecular starter kit had already been assembled somewhere else entirely, long before Earth ever had an ocean to hold it, and simply arrived ready-made, carried down from somewhere out beyond our own atmosphere? It was an idea that, on its face, seemed only to relocate the mystery rather than solve it, since something, somewhere, would still have needed to build those ingredients in the first place. But it also offered something Darwin's pond could not, an entirely different set of raw materials, chemistry forged and tempered under conditions no laboratory on Earth could easily recreate, and quite possibly a great deal more time in which to do the forging. The man who first asked that question out loud, in language serious enough for other scientists of his day to take notice, was a Swedish chemist named Svante Arrhenius, and it is worth understanding exactly who he was before we get to what he actually proposed, because he was no crank standing at the edge of respectable science, no dreamer with an idle theory and nothing to back it up. He was, by the turn of the 20th century, one of the most decorated and widely respected chemists alive anywhere in the world. Born in 1859 near the old university town of Uppsala, Sweden, the son of a land surveyor, Arrhenius reportedly taught himself to read years before he ever set foot in a schoolroom, simply by watching his father work through columns of figures in a ledger, the sort of early, restless curiosity that would define the rest of his life. He had built his early scientific reputation on a discovery that still sits at the foundation of modern chemistry classrooms, an explanation of how ordinary table salt and countless other compounds like it break apart into charged particles the moment they dissolve in water, quietly carrying electrical current through a solution that otherwise looks like nothing more than plain water in a glass. His path to recognition for that idea, however, was anything but smooth. When Arrhenius first submitted a dissertation containing this very theory to his examiners at Uppsala in 1884, they were unconvinced and awarded it only the lowest passing grade the university allowed, effectively closing the door on any comfortable academic career in Sweden itself. It was only when he mailed copies of his work to chemists elsewhere in Europe, men like Wilhelm Ostwald in Germany and Jacobus van 't Hoff in the Netherlands, that its true significance was recognized. Both men would go on to become towering figures of chemistry in their own right, and both credited Arrhenius's dismissed dissertation as foundational to their own later work. Within 2 decades, the same insight his own professors had barely tolerated carried him all the way to Stockholm, where he collected the Nobel Prize in Chemistry in 1903. By every account, he was also an unusually warm and sociable man for a scientist of his standing, known for his lively lectures and his wide circle of friends and correspondents scattered clear across Europe and the United States. He was, in every sense, a serious man doing serious science, the sort of figure whose photograph belonged in a chemistry textbook long before it belonged beside any headline about life arriving from another world. But Arrhenius had a restless, wide-ranging mind that refused to stay confined to any single discipline. That same curiosity had already led him, a few years earlier, to work out one of the first serious scientific explanations for how carbon dioxide in the atmosphere traps heat near a planet's surface, laying early groundwork for what we would today call climate science. In the years surrounding his Nobel Prize, that same restless attention increasingly drifted upward, away from beakers and dissolved salts, and toward the physics of the cosmos itself. Among the many puzzles that caught his interest was a genuinely strange observation that astronomers had spent centuries turning over without ever fully explaining, one that the astronomer Johannes Kepler himself had puzzled over some 3 centuries earlier. The mystery had captured imaginations for centuries by then, since a comet's tail is one of the most striking sights the naked eye can witness in the night sky, a pale, curving plume, sometimes stretching wider across the heavens than an outstretched hand held at arm's length. Anyone who has watched a comet cross the night sky, whether through a telescope or in an old photographic plate pinned up in an observatory office, will notice something peculiar about its glowing tail. That tail never simply trails behind the comet the way smoke trails behind a moving locomotive. Instead, it always points away from the sun, no matter which direction the comet itself happens to be traveling at that moment, sometimes even appearing to stretch out ahead of the comet as it pulls away from its closest approach. The answer, confirmed experimentally only a handful of years before Arrhenius took up the question in earnest, turned out to be that ordinary sunlight itself, the very same light that warms a front porch on a summer afternoon, carries within it a faint but entirely real physical push. Separate laboratories, one in Russia and another in the United States, had each managed to demonstrate, through careful and painstaking experiments, that a beam of light striking an object exerts a small but genuinely measurable pressure on it, gently nudging that object in the direction the light happens to be traveling. Bathed in enough sunlight for long enough, even something as insubstantial as the fine dust drifting in a comet's tail could be pushed steadily outward, away from the star that lit it in the first place, which is exactly why that tail always points away from the sun, rather than trailing obediently behind. Arrhenius took that discovery and, in the way that only a truly restless mind can, asked a far larger question of it. If the pressure of ordinary starlight could push fine dust across millions of miles within the confines of our own solar system, why should it not, given enough patience and enough time, push something small enough across the far greater emptiness that separates one star system from its neighbors? He proposed that living things, reduced to their simplest and hardiest possible form, the tough, dormant spores produced by certain bacteria and other simple organisms right here on Earth, might be light enough and durable enough to survive exactly that kind of impossibly long journey. None of this was idle fantasy on his part. Spores produced by certain bacteria really are extraordinarily tough by the standards of any other living thing on this planet. They can withstand years of drought, extremes of heat and cold that would kill the active organism outright, and long stretches with no food or water whatsoever, simply waiting, sometimes for a very long time indeed, until conditions improve enough to stir back into active life. If any living thing on Earth seemed built to survive a genuine ordeal, a spore seemed like a reasonable candidate. In laboratory tests, some bacterial spores have been revived after decades spent sealed away in dry storage, and researchers have reported far older specimens recovered from ancient amber or deep permafrost that may have lain dormant far longer still. Although such extreme claims remain difficult to fully verify and are treated cautiously within the scientific community, lifted somehow off the surface of a world and carried above its atmosphere, such a spore might drift outward for years, then centuries, then longer still, nudged along the entire way by the endless, patient pressure of starlight, until it happened, purely by chance, to drift close enough to some other world to fall into its atmosphere, and, if the conditions there allowed it, begin the whole long business of life all over again on entirely new ground. He gave the idea a name built from old Greek roots, meaning, roughly, seeds everywhere, and that name has outlived nearly everything else about his original proposal, panspermia. Not every scientist of his era was persuaded, even at the time. Some colleagues raised early doubts about whether any living thing, however hardy, could truly endure the specific hazards of open space for the enormous amount of time such a crossing would require. Arrhenius himself was aware of these objections and answered them as best contemporary science of his day allowed. But the full picture of just how hostile that environment truly is would not come into focus for scientists until long after his own lifetime. Once humanity finally possessed the instruments needed to measure that hostility directly, rather than simply guess at it from the ground, he laid the idea out formally, for the first time in 1903, tucked inside a dense, 2-volume scientific work titled, in English, Textbook of Cosmic Physics, written for an audience of fellow scientists rather than the general public. 5 years later, in 1908, he returned to the idea in a far more accessible book meant for ordinary readers, one that appeared in English translation under the title Worlds in the Making. That second book found an audience far beyond the walls of any laboratory. In Sweden, it reportedly outsold every previous work of natural science ever published there, a genuine sensation among readers who had likely never before given serious thought to where the story of life on Earth might have actually begun. Arrhenius wrote with the confident, expansive voice of a man thoroughly convinced he had identified something real, a mechanism grounded in demonstrated, tested physics rather than mere wishful speculation, by which life might hop quietly between worlds, much the way dandelion seeds drift across an open summer field on nothing more than a passing breeze. There was, however, a serious problem lurking inside Arrhenius's otherwise elegant idea, one that other scientists would need several more decades to fully appreciate, and it had nothing whatsoever to do with the physics of light pressure itself, which was solid, well-tested, and entirely correct as far as it went. The problem was the environment any such spore would actually have to survive along the way. The space between the stars is not simply empty and cold and still. It is bathed, constantly and without any let-up, in radiation of exactly the kind that shreds the delicate molecules living things depend upon to function at all, breaking apart the very chemical bonds that hold genetic material together. Even here, so close to home, the danger is real enough that it commanded the world's attention within Arrhenius's own lifetime. When the United States launched its first satellite in 1958, the physicist James Van Allen discovered that Earth itself sits wrapped in wide, invisible bands of trapped radiation circling the planet, bands strong enough that engineers have had to plan every crewed mission since around, threading a careful path through and beyond them. Astronauts who traveled to the moon during those years reported something the engineers back home had not entirely anticipated. With their eyes closed in the darkness of their spacecraft, they would occasionally see faint flashes and streaks of light, phantom visions with no earthly source. Those flashes, scientists later confirmed, were caused by individual particles of cosmic radiation passing directly through the fluid of the astronaut's own eyes, a small, personal demonstration of exactly the kind of unfiltered, energetic bombardment that fills the space between worlds, let alone the far vaster space between stars. If a comparatively thin, well-mapped band of radiation held close by Earth's own magnetic field demands that much caution, it is hard to imagine a dormant spore surviving, unshielded and alone, a crossing between two entirely different star systems, a journey that even at the fastest speeds starlight pressure could plausibly manage would very likely stretch on for many thousands of years, adrift the entire time in the full, unfiltered radiation of open space. That particular difficulty would not become fully clear to the scientific world for several more decades after Arrhenius first set his idea down on paper, and by the time it did, the conversation about life traveling between worlds had already begun to change shape entirely, carried forward now by scientists working with tools that a chemist at the turn of the 20th century could scarcely have imagined. Powdered fragments of actual meteorites, waiting patiently in museum drawers for the right instruments to be invented. Chemical traces lifted directly from the glowing tail of an actual active comet by an orbiting spacecraft. And, eventually, astonishingly, sealed capsules of dust carried home across millions of miles from asteroids by robotic explorers built and launched by human beings, in countries on opposite sides of the world working, for once, toward the very same question. What those tools would go on to reveal was not quite the idea Arrhenius had proposed. The travelers themselves would turn out to be far smaller and far humbler than any spore, small enough that no one would ever need to ask whether they were alive to begin with, and yet, as later observers would come to appreciate, resting on a great deal more solid evidence than his original vision of living spores drifting patiently between the stars ever managed to gather on its own. That shift, from a story about living things crossing the stars to a story about the molecules those living things are built from, would take the better part of a century to unfold, and it would lead scientists to some of the most unlikely places imaginable. A scorched patch of the Australian outback, the tail of a comet racing past the sun, and, eventually, small, dark asteroids drifting quietly through the space between Earth and Mars. Powdered fragments of actual meteorites were the first of those unlikely places, and the story of the most important one among them begins not in a laboratory at all, but in a quiet stretch of countryside in Victoria, Australia, on an otherwise unremarkable September morning in 1969. Barely 2 months earlier, the whole world had watched 2 American astronauts step out onto the dust of the moon, the culmination of a decade spent reaching outward from Earth with every resource a nation could muster. And now, with almost no fanfare at all, something had arrived from much farther away than the moon, entirely uninvited, streaking across the Australian sky in a fireball bright enough to be seen and heard for miles before it broke apart high in the atmosphere, scattering a literal rain of blackened stones across the fields, rooftops, and quiet backyards surrounding the small township that would go on to lend the object its name. Local residents gathered pieces of it in the days that followed, some of them still faintly warm to the touch, having no idea they were holding something that would occupy chemists for the rest of the century and beyond. Word of the fall reached scientists quickly, and within weeks, a team of chemists working at one of NASA's own research centers had begun testing fragments of the rock, curious whether a meteorite might hold any trace of the same chemistry that builds living things here on Earth. It was, in its own quiet way, an extraordinary stroke of good timing. Murchison was a large fall, generous enough that more than 200 pounds of it were eventually recovered, which meant there was enough material to share freely among laboratories, to spend generously on early, imperfect tests, and still have plenty left over decades later for instruments nobody in 1969 could have anticipated. The Murchison meteorite, as it came to be known, belongs to a category of space rock called a carbonaceous chondrite, a name that simply means it is rich in carbon and built from some of the oldest, least altered material left over from the very birth of the solar system, dated by later analysis to roughly 4.5 billion years old, very nearly as old as the Sun itself. Meteorites of this particular kind are genuinely rare finds. The great majority of meteorites that reach Earth are stony or metallic fragments knocked loose from asteroids that were, long ago, melted and reshaped by heat and pressure, their original chemistry cooked away in the process. And only a small minority of all recorded falls belong to the carbon-rich family Murchison represents. Carbonaceous chondrites like Murchison are treasured precisely because they mostly skip that violent reprocessing, preserving instead the raw material the solar system itself was assembled from, largely undisturbed since before Earth even existed, a fragment of that same restless, bombarded era from earlier in this story, only preserved rather than melted down and lost. When those first chemists began examining the freshly fallen fragments, they found something that made headlines well beyond the world of geology. Genuine amino acids, the same humble building blocks of protein that link together inside every living cell on this planet, embedded directly in the meteorite's interior. Scientists had good reason to move carefully with a claim like that. Just a few years earlier, a much older meteorite called Orgueil, which had fallen in France back in the 1860s and had long been preserved in a French museum, became the center of an embarrassing scandal when one preserved sample turned out to contain a lump of ordinary coal, along with what looked like a deliberately embedded seed pod, clear evidence that someone, at some point over the intervening century, had tampered with the specimen to manufacture false proof of extraterrestrial organic matter. The fraud was exposed publicly, and the damage it did to the field's credibility was real. It meant that when Murchison fell only a handful of years later, carrying what appeared to be genuine amino acids of its own, researchers understood perfectly well that sloppy, hasty, or overeager claims would not be forgiven a second time, and they treated every fragment accordingly, working only with freshly broken interior pieces that had never been touched by bare hands, in conditions designed to keep the meteorite's own chemistry as isolated from the surrounding laboratory as possible. That announcement immediately raised an obvious and entirely reasonable objection, one that any careful scientist would have raised, and one the researchers studying Murchison had to answer convincingly before anyone would take the finding seriously. A rock that spends any time at all sitting in an open field, surrounded by soil, insects, and the countless microorganisms that blanket nearly every surface of this planet, is not exactly a clean laboratory sample. How could anyone be sure these amino acids had not simply soaked in from the Australian dirt itself, or been introduced by careless handling after the fact, rather than having ridden inside the rock the entire way from space? For years afterward, skeptics within the scientific community pressed exactly this point, and it took patient, repeated testing on fragment after fragment, laboratory after laboratory, before the case was considered settled. The answer came from two independent lines of evidence, each of which pointed in the same direction. The first involves a property of amino acids called handedness. Nearly every amino acid used by life on this planet can, in principle, form in one of two possible mirror image shapes, much the way a left-hand glove and a right-hand glove are identical in every measurable respect, except that one will never sit comfortably. On the other hand, life here overwhelmingly uses only the so-called left-handed version of these molecules, almost without exception, in every plant, animal, and microbe that has ever been examined, as though every household on Earth kept a drawer full of left-hand gloves and nothing else, generation after generation, without a single right-handed one ever turning up by mistake. If Murchison's amino acids had simply soaked in from surrounding Earth life, they should have shown that same overwhelming left-handed preference. Instead, chemists found something quite different, a mix of left-handed and right-handed forms in close to equal proportion, the signature you would expect from ordinary, non-living chemistry, rather than from any biological source, earthly or otherwise. The second line of evidence came from isotopes, the very slightly different weights that atoms of the same element can carry depending on how many neutrons sit inside their nucleus, a kind of quiet fingerprint stamped onto a molecule by the particular conditions under which it formed. Carbon and hydrogen from Murchison's amino acids carried isotope ratios distinctly different from anything found in ordinary Earth biology, including a heavier form of hydrogen called deuterium, present in noticeably different proportions than it is in Earth's own water and organic matter, pointing toward formation in an environment quite unlike our own planet's surface, likely in the frigid outer reaches of the solar system, or in the cold clouds of gas and dust that came together to form it. Taken together, these two clues told chemists that the amino acids inside Murchison were not contamination at all. They were original passengers, older than the rock's own arrival on Earth, quite possibly older than Earth itself. There was a further, quieter clue buried in Murchison's chemistry that took even longer to appreciate fully. Alongside the familiar amino acids that make up proteins in every living thing on this planet, the meteorite also contained a whole catalogue of unusual amino acids that no known form of terrestrial life uses at all. Among them one called isovaline, a molecule that has never once turned up as a building block of any protein in any living organism ever studied, yet sat there in the meteorite in meaningful quantity, in that same telltale 50-50 mix of mirror image shapes. Its presence was, in its own way, some of the strongest evidence of all. It is one thing to imagine ordinary biological contamination sneaking a handful of familiar molecules into a rock. It is quite another to imagine it inventing entirely new ones that appear nowhere in Earth's own biology, and then depositing those alongside the familiar ones in just the proportions non-living chemistry would be expected to produce, and in no proportion any earthly contaminant ever would. Researchers have since catalogued dozens of these unfamiliar amino acids across Murchison and meteorites like it, an entire alternate alphabet of molecules that simply never made the cut when life on Earth settled billions of years ago, on the particular set of building blocks it still relies on today. Over the decades that followed, laboratories around the world kept returning to the same modest supply of Murchison fragments again and again as their instruments grew steadily more sensitive. What had first been a fairly short list of confirmed molecules back in the early 1970s grew into hundreds of distinct organic compounds by the time researchers were done, all pulled from a single rock that had been sitting quietly in museum drawers and laboratory freezers the entire time, waiting patiently for technology to catch up with it. Murchison, in time, became something of a gold standard among meteorite researchers. The sample every new instrument and every new extraction technique was tested against first, simply because so much of it had fallen, and because scientists already understood its chemistry better than that of almost any other rock ever recovered from space. A curious fragment of stone, gathered by farmers who had no training in chemistry at all, ended up outlasting the careers of the scientists who first studied it, still yielding fresh discoveries to researchers who had not yet been born the September morning it fell. In many ways, the very same broad question Miller and Urey had once tried to settle inside a single sealed flask on a laboratory bench back in 1952 was now being answered again, independently, out of a rock that had simply fallen out of the sky on its own. Amino acids, however, are only half of what a complete genetic alphabet requires, and for a very long time, the other half of that alphabet proved far more stubborn to find inside Murchison. Life's instructions are written using 5 distinct nucleobases, 2 slightly larger molecules called purines, and 3 smaller, more delicate ones called pyrimidines. And, for decades after Murchison's fall, chemists, using the extraction methods available to them, could only coax 3 of those 5 out of the meteorite with any confidence: 2 purines and just 1 of the 3 pyrimidines. The 2 remaining pyrimidines were considerably more fragile than their purine cousins, prone to breaking down entirely when exposed to the scalding hot acid researchers had long relied on to pull organic material out of powdered rock in the first place. For decades, nobody could say for certain whether those 2 missing molecules had simply never formed in the meteorite's parent body at all, or whether they had been there the entire time, quietly destroyed by the very method meant to reveal them. An uncomfortable kind of uncertainty for a rock that had already given up so many of its secrets. The missing pieces finally turned up in 2022, when a research team led by the chemist Yasuhiro Oba tried a considerably gentler approach, soaking crushed samples of the meteorite in cool water rather than scalding acid before running the resulting solution through sensitive modern instruments capable of detecting even the faintest trace of a target molecule. Where the old, rough method had likely been destroying the 2 missing pyrimidines before anyone could ever measure them, the new, patient method finally succeeded in drawing them out intact. After more than 50 years, the full set of 5 nucleobases had at last been confirmed inside a single meteorite. The same 5 molecular letters used to write every strand of genetic material in every living thing on this planet, sitting quietly inside a rock that had fallen from the sky the same year human beings first walked on the moon. As compelling as Murchison and meteorites like it had become, though, they carried one limitation that no amount of clever chemistry could fully erase. However careful the analysis, however convincing the case built from handedness and isotopes and unfamiliar molecules, a meteorite is, by definition, an object that has already survived a fiery plunge through Earth's atmosphere and then sat exposed on the ground, however briefly, before anyone recovered it. Scientists wanted something cleaner still, a sample of this same primordial chemistry examined at its actual source, out in space, before it had ever come anywhere near Earth's dirt, Earth's air, or Earth's own busy biology at all. That ambition led, remarkably, to 2 entirely different spacecraft, launched by 2 different space agencies, both aimed at the same deceptively simple question, and both arriving at their answers within the same broad stretch of years. The first of these was a NASA mission named Stardust, launched in 1999 and sent on a multi-year journey to rendezvous with a comet called Wild 2. Wild 2 itself was a modest potato-shaped chunk of ice and rock only a few miles across, its surface pocked with craters and cliffs that spacecraft cameras revealed in surprising, almost geological detail as Stardust made its approach. Rather than landing or orbiting, Stardust was built to fly directly through the comet's coma, the loose, glowing cloud of gas and dust that surrounds an active comet's icy core, and to catch a sample of that material as it passed through at a relative speed several times faster than a rifle bullet. Fast enough that any conventional collection material, anything solid and dense, would have vaporized the dust on contact just as thoroughly as it destroyed itself. The solution engineers settled on was a strange, almost otherworldly substance called aerogel, a silica-based material so remarkably light and full of empty space that people who have handled it often describe it as looking like solid smoke frozen in place. When tiny dust grains from the comet struck the aerogel in early 2004, they burrowed gently to a stop deep inside it without being destroyed by the impact, leaving slender, carrot-shaped tracks that scientists could later follow, like tiny breadcrumbs, straight back to the particles responsible, a feat of careful engineering built around a single, narrow window of opportunity that would never come again. Stardust returned that collector safely to Earth in 2006, and the sheer volume of aerogel waiting to be examined turned out to be more than any single research team could manage alone. NASA eventually turned to the public for help, inviting ordinary volunteers from around the world to examine digital microscope images of the aerogel from their own home computers, searching patiently for the telltale tracks left behind by genuine comet dust among countless false alarms caused by ordinary dust and scratches. Tens of thousands of these volunteer searchers took part, and among the compounds researchers eventually identified in the particles those searchers helped uncover was glycine, the simplest of all amino acids, caught directly inside material that had come from a comet rather than an asteroid or a meteorite that fell through the atmosphere unsupervised. Confirmed in 2009, the glycine carried an isotope signature distinct from anything found in ordinary Earth chemistry, the very same kind of chemical fingerprint that had helped settle the contamination question for Murchison years earlier. For the first time, scientists held a piece of a comet in hand, gathered directly from its source, and it too carried the building blocks of protein. In that very same year that Stardust threaded its way through the tail of Wild 2, a second spacecraft was leaving Earth on a far longer and more patient errand. The European Space Agency's Rosetta mission launched in 2004 as well, though its destination, a modest, oddly shaped comet named 67P Churyumov-Gerasimenko, lay so far off in its orbit that reaching it would take Rosetta the better part of 10 quiet years of travel through the solar system, looping past Earth 3 separate times and past Mars once, borrowing a little speed from each flyby, the way a skater gathers momentum from a series of long, patient turns around a rink, since no rocket built at the time could carry enough fuel to make that crossing in a straight line. Each flyby had to be aimed with extraordinary precision years in advance, the spacecraft threading a gravitational needle it would not get a second chance to thread before finally arriving anywhere close to its actual destination. So long a journey that mission engineers eventually put the spacecraft into a deep, dormant hibernation for more than 2.5 of those years simply to conserve power, and then had to hold their breath in early 2014 while they waited, without any signal at all, to learn whether Rosetta would faithfully wake itself back up on schedule, out near the orbit of Jupiter, exactly as it had been programmed to do so many years before. It did, and by the late summer of that same year, Rosetta had settled into orbit around the comet's nucleus, close enough to send back images showing a strange, dark, two-lobed shape that reminded more than one member of the mission team of a rubber duck floating in space. That November, the mission released a small lander named Philae down toward the surface, And though its anchoring harpoons failed to fire, and it bounced awkwardly across the comet's low gravity before finally coming to rest in an awkward, shadowed spot, the little lander kept working for some 60 hours regardless, sending back photographs from the surface of an active comet before its battery finally ran down in the shadow where it had come to rest. And the drama of that landing captured public attention around the world, becoming, for a little while, front-page news everywhere from Europe to the United States. Among the instruments Rosetta carried in orbit above that scene was a sensitive mass spectrometer named Rosina, designed to sniff out and identify individual molecules drifting in the thin cloud of gas that a comet sheds as sunlight slowly warms its icy surface. In 2016, the scientist leading that instrument's team, Catherine Altwegg of the University of Bern, announced what her group had found drifting in that gas. Glycine once again, detected directly and repeatedly in the material, actively escaping from the comet's surface alongside two related molecules known to be its chemical precursors. Altwegg called it the first unambiguous detection of an amino acid ever made directly at a comet, a meaningful distinction from Stardust's returned grains, since this glycine had never touched a container, an aerogel block, or Earth's atmosphere at all. It had simply been measured exactly where it drifted, out in space, by an instrument riding alongside the comet itself. Glycine, it turns out, is a genuinely useful molecule for this particular kind of detective work, precisely because of how stubbornly it resists turning from a solid into a gas. It generally needs to warm past roughly 300 degrees Fahrenheit before enough of it will vaporize to be detected by an instrument like Rosina, a threshold that only a narrow stretch of a comet's surface reaches, and typically only when the comet has swung in close enough to the sun to feel real warmth for the first time in years. That same stubbornness is precisely why Rosetta's team also found the gas rich in dust at the moments of strongest detection, since the glycine appeared to be riding out to freedom, clinging to grains of warming dust rather than sublimating cleanly on its own. Alongside the glycine, Rosina also picked out phosphorus in the comet's thin atmosphere, an element every living thing on Earth depends on for the backbone of its genetic material and for the membranes that hold its cells together, an element without which the whole architecture of a living cell has no way to hold its shape, and this marked the first time that particular element had ever been confirmed drifting freely around an active comet. One more piece of life's basic hardware, turning up in a place no one could accuse of having been contaminated by anything Earth ever produced. None of this had been the work of any single nation acting alone. Australian farmers had gathered the first fragments. American chemists had run the first tests. A Japanese-born chemist, working decades later, found the meteorites' missing molecules using a technique refined half a world away from where the rock had actually fallen. An American spacecraft had chased down one comet while a European one chased down another, and ordinary volunteers scattered across dozens of countries had helped comb through the results by hand, from their own kitchen tables, simply because they found the question worth their time. Whatever this chemistry turned out to mean, it had already become a thoroughly international undertaking, pieced together in fragments by hands that had never met, working toward the very same distant answer. Taken all together, the picture assembled from these two very different missions, working on two very different comets, using two entirely different methods, told an unusually consistent story. One comet, sampled by catching its dust as a spacecraft streaked past. A different comet, sampled by orbiting patiently alongside it for months and sniffing its escaping gas the whole while. Both, independently, turned out to carry glycine, the same amino acid found decades earlier, soaked into a fallen meteorite half a world away in the Australian countryside. Whatever process builds these molecules out in the cold and dark between the planets did not appear to be a rare accident confined to one unlucky rock or one unusual comet. It looked, increasingly, like an ordinary and perhaps quite common feature of the material the solar system itself had been assembled from in the first place, repeated again and again across wildly different objects, using wildly different methods of detection, and still arriving at the very same answer. By the years following Rosetta's mission, a meaningful share of the scientists who studied the early solar system for a living had quietly come around to treating this delivery of raw ingredients not as some exotic long shot, but as an ordinary and expected part of how a young planet accumulates its chemistry. None of this, on its own, solved the timing problem described earlier in this story, the uncomfortably short window between a bombarded, barely cooled young Earth and the appearance of its first confirmed life. But it did offer that problem a genuine, evidence-backed way forward. If a meaningful share of the raw chemical ingredients life would eventually need had already been forming out in space, in comets, and in the parent bodies of meteorites, long before Earth's surface had even finished settling down, then early Earth would not have needed to build every last piece of that chemistry entirely from scratch, entirely on its own, entirely within that narrow window. It would only have needed to receive a head start, carried down out of the sky, and then find a way to put that head start to use. And yet even this remarkable convergence of evidence, meteorite and comet alike, still left a door open for the most stubborn kind of doubt. A meteorite, however carefully its handedness and isotopes are measured, has still fallen through an atmosphere and touched the ground before anyone examined it. A comet's coma, however precisely an orbiting instrument measures it, is still gas and dust, drifting loose in space, sampled indirectly and from a distance rather than held, sealed, and carried home intact for a full and leisurely study on Earth. What scientists wanted next was something neither a fallen rock nor a passing wisp of gas could offer, an actual, solid piece of a pristine, ancient body gathered directly at the source by a spacecraft built for exactly that purpose, sealed away before it ever touched Earth's air even once, and carried home not by accident, the way Murchison had arrived, but entirely on purpose, in a capsule built and aimed by human hands for that very reason. A meteorite tells you what survived the fall. A comet's gas tells you what happened to escape into the light. Only a sealed, deliberate sample could tell scientists what a whole world, untouched and unweathered, actually looked like on the inside. And by the years following Rosetta's long vigil at its comet, 2 different space agencies on 2 different continents were already building the spacecraft that would attempt exactly that, aimed not at a comet at all this time, but at something smaller, darker, and far quieter, tumbling patiently through the space between the orbits of Earth and Mars. The 2 spacecraft in question turned out to be aimed at 2 separate worlds, each barely large enough to qualify as a world at all, tumbling quietly through the space between Earth and Mars long before either agency ever thought to visit them. One belonged to Japan's space agency, the other to America's, and though the 2 teams worked independently on opposite sides of the Pacific, they were reaching for the very same prize, an untouched, uncontaminated sample of the same ancient material that had already turned up again and again in meteorites and comets alike. Neither team was starting entirely from scratch, and neither mission would prove easy. Both would spend the better part of a decade planning, building, launching, and waiting all for a handful of minutes of actual contact with their respective targets, the kind of patient, deferred payoff that space exploration seems to specialize in more than almost any other human undertaking. Japan's spacecraft, whose name translates loosely to Falcon, launched in 2014, bound for a small, dark asteroid called Ryugu, drifting in an orbit that carries it back and forth across Earth's own path around the Sun. Space agencies rarely get a clean first attempt at anything this difficult, and Japan's own history with asteroid sample return proved the point especially well. It was not Japan's first attempt at this particular feat. An earlier mission, sent years before to a much smaller asteroid named Itokawa, had suffered one technical setback after another on its way there and back, losing engines, losing contact with Earth for weeks at a time, and ultimately returning home with only the barest trace of dust rather than the generous sample its engineers had hoped for. That first mission became, in its own quiet way, a source of national pride precisely because it had barely survived at all, and its successor was built from the start to fix everything that had gone wrong the first time around. That first spacecraft had lost 2 of its 4 ion engines along the way, gone silent for nearly 7 weeks after a fuel leak sent it tumbling out of control, and very likely fired its sampling mechanism incorrectly when it finally reached the surface, leaving engineers uncertain for years afterward whether it had collected anything at all. It came home 3 years later than planned, its heat shield glowing as it broke apart deliberately in the upper atmosphere during a final, fiery return, while a small capsule beneath it parachuted safely down to the Australian desert carrying, against every reasonable expectation, a pinch of genuine asteroid dust. The story of that near failure and its narrow, stubborn success became beloved across Japan, inspiring documentaries and feature films of its own, and it left the engineers of its successor with a long, hard-earned list of everything that needed to work better the second time around. Neither Ryugu nor Bennu had been chosen at random. Long before either spacecraft ever left the ground, astronomers had studied both asteroids from Earth using ordinary telescopes, examining the particular way each one reflected sunlight across different wavelengths, a kind of remote chemical signature that let scientists guess, with reasonable confidence, which asteroids out of the many thousands catalogued in the solar system were likely to be dark, carbon-rich, and largely unaltered since the solar system's earliest days, rather than one of the far more common types that had already been baked and reshaped by billions of years of heat and collision. Only after that careful, earth-bound screening did either agency commit years of planning and enormous expense to actually flying out and confirming the guess up close. After a 4-year cruise through the solar system, the second Hayabusa arrived at its target in the summer of 2018, and the images it sent home surprised even the mission's own scientists. Ryugu turned out to be shaped almost like a spinning top, or a rough, dark diamond, a little more than half a mile wide, its surface a jumble of boulders rather than the smoother, dustier world many had expected. Rather than landing outright, the spacecraft approached the surface in a series of brief, careful touches. On its first descent, it fired a small metal projectile into the asteroid to kick up material from just beneath the surface, catching the resulting spray of dust and pebbles in a horn-shaped collector mounted beneath its own body. Then, in a maneuver with no real precedent, the mission released a separate device entirely, a small copper impactor that blasted a fresh, artificial crater into Ryugu's surface from a safe distance, exposing material that had never once been touched by the sun's radiation or the thin wash of charged particles that constantly bombard an airless world. The spacecraft returned days later to collect a second sample from inside that freshly exposed crater, gathering material even more pristine than what its first touchdown had managed to reach. Material that had spent the last several 100 million years shielded from the same slow, steady weathering that every exposed surface in the solar system endures. The mission turned for home in the closing weeks of 2019, releasing a small return capsule that streaked back through Earth's atmosphere and touched down in the South Australian outback in December of 2020, not so very far, in the end, from the same countryside where Murchison had fallen on its own, uninvited, half a century earlier. Inside the capsule sat just over 5 grams of Ryugu, a quantity smaller than a single sugar packet, carried afterward to a dedicated curation facility built specifically to house it in Sagamihara, Japan, where it has been portioned out to research teams around the world in careful, minuscule amounts ever since. Facilities like this one exist for exactly the reason 100 years of meteorite skepticism might suggest: to make the handedness and isotope arguments unnecessary in the first place, by never letting the sample near ordinary Earth air or human skin at all. Technicians handle the grains inside sealed glove boxes filled with pure nitrogen rather than the ordinary air of the room around them, using tools clean to a standard closer to what a hospital operating theater might expect than an everyday laboratory, so that whatever a chemist eventually finds inside a single grain of asteroid dust can be trusted from the very first measurement as belonging to the asteroid and to nothing else, rather than requiring the same painstaking after-the-fact detective work that handedness and isotopes once had to perform for Murchison. Among the researchers who eventually turned their instruments toward that small supply of asteroid dust was the same chemist whose gentler, cool water extraction technique had finally coaxed the last 2 missing nucleobases out of Murchison only months before Ryugu's sample reached Earth. Yasuhiro Oba and his colleagues applied that same patient method to the Ryugu grains, and in 2023 they announced they had found uracil, one of the 5 molecular letters used to write genetic instructions, sitting inside material gathered directly from an asteroid rather than pulled from a meteorite that had first survived a fiery plunge through the atmosphere. Alongside the uracil sat niacin, a compound better known today as a common vitamin, the same one added to enriched bread and breakfast cereal on grocery shelves across the country. One more piece of ordinary, everyday biological chemistry turning up in a sample that had never touched Earth's surface, even once before. Scientists opened its capsule under carefully controlled conditions, built to keep any stray earthly contamination out entirely. The same grains also carried clay-like minerals that only form in the presence of liquid water, evidence that Ryugu's parent body, whatever larger asteroid it broke away from long ago, once held pockets of liquid water somewhere inside it, slowly altering its original minerals over long stretches of time before a collision shattered that parent body and left Ryugu itself as one of the scattered pieces, tumbling on its own ever since. That announcement alone would have been remarkable enough, but it was only the opening chapter of Ryugu's story. Uracil is only one of the 5 nucleobases, and for a while, the fate of the remaining 4 in this particular sample remained an open question, the same kind of question that had taken more than 50 years to resolve for Murchison. The answer came only this past spring, when Oba, working now alongside colleagues including the chemist Toshiki Koga, announced in a study published in Nature Astronomy that the full set of all 5 canonical nucleobases had at last been confirmed inside the Ryugu grains. The complete molecular alphabet needed to write the genetic instructions of every living thing on this planet, recovered from a handful of dust gathered by a robotic arm on a tumbling, sunless rock nearly 200 million miles from home. It had taken chemists more than half a century to draw that same complete set out of a meteorite that fell into an open field. It took barely 3 years to do the same for a sample that had been sealed away deliberately from the very start. While the second Hayabusa was making its own careful approach to Ryugu, a separate spacecraft, built and flown by NASA, was closing in on a different asteroid entirely. This mission, known by the rather elaborate name Origins, Spectral Interpretation, Resource Identification, Security, Regolith Explorer, or more simply as OSIRIS-REx, launched in the fall of 2016, aimed at a somewhat smaller, similarly dark body named Bennu, an asteroid a little under a third of a mile across in its own right, roughly half the width of Ryugu, though shaped, in an odd coincidence, much like it, another rough, spinning top, tumbling quietly through space. Bennu, too, turned out to be more rugged than mission planners had hoped, its surface strewn with boulders large enough to threaten a careless landing, forcing the team to abandon their original plan of choosing among several wide, sandy-looking patches, and instead aim for a far smaller, more cramped target, barely larger than a few parking spaces laid side by side. The mission's lead scientist, Dante Loretta of the University of Arizona, had spent years preparing for a single, carefully rehearsed encounter with the asteroid's surface. And when that moment finally arrived in October of 2020, it did not go quite the way anyone on the team had expected. Bennu's surface, mission planners had assumed from years of remote observation, would behave something like a gravel driveway, firm enough to support the brief weight of a spacecraft's touch. The sampling arm itself, nicknamed for the acronym describing its own mechanism, extended slowly downward over several tense hours before finally making contact, firing a short burst of nitrogen gas meant to stir up loose material into its collector head during the mere handful of seconds the whole maneuver was designed to last. Instead of meeting firm resistance, the collector sank down through the surface far deeper than engineers had ever planned for, as though the asteroid were built more like a bank of loose, dry snow than solid rock. And cameras, watching the encounter, showed a small storm of gravel and dust kicked up around the spacecraft, some of it drifting off into space entirely rather than settling back down under Bennu's famously feeble gravity. In those few frantic seconds, the collector gathered so much material so quickly that some of it became lodged in the flap meant to seal the sample safely away for the long trip home. Rather than risk losing any of that unexpectedly generous haul to the vacuum of space, the team made the decision within days to skip several planned checks and stow the sample immediately. A small, tense drama, playing out entirely through radio signals, crossing millions of miles of empty space, watched over anxiously by engineers back on Earth, who could do nothing in the moment but wait for confirmation that the sample chamber had closed successfully. That moment of good fortune paid off handsomely. When the return capsule finally touched down in the Utah desert in September of 2023, it carried a little over 121 grams of Bennu, more than 20 times the modest haul the second Hayabusa had brought back from Ryugu only a few years earlier, making it, by a wide margin, the largest sample of an asteroid ever delivered safely into human hands. True to the same spirit of international cooperation that had already carried this story across 2 continents, NASA set aside a small portion of the Bennu sample for Japanese researchers to study in exchange for a matching portion of Ryugu so that scientists on both sides of the Pacific could run their own instruments on both asteroids, rather than relying solely on results reported second-hand from the other side of the world. Much of that material now resides in a specially built curation laboratory at Johnson Space Center in Texas, portioned out carefully to research teams in much the same deliberate way. Ryugu's own small supply is managed half a world away. Both agencies, in fact, deliberately held most of each sample back rather than handing it all over to researchers at once, setting aside the majority of the material in long-term storage for scientists not yet born, working with instruments not yet invented, the same patient logic that had eventually paid off so handsomely for Murchison decades after it first fell. The spacecraft that gathered Bennu's sample, meanwhile, was not finished with its working life once the capsule had parachuted safely into the Utah desert. Renamed OSIRIS-APEX and redirected toward a fresh assignment, it is now making its way toward a much larger asteroid named Apophis, one that will pass closer to Earth than many communications satellites orbit when it swings by in 2029. And mission planners intend to have the same well-traveled spacecraft waiting there to watch the encounter up close, examining how the strain of Earth's own gravity reshapes an asteroid's surface in real time, a fitting second act for a spacecraft that had already learned so much from a single asteroid surface once before. More than a year passed between the moment that capsule touched down in the Utah desert and the moment its full findings finally reached the public, a stretch of careful, unhurried laboratory work that involved dozens of scientists across multiple countries, each running their own tests on their own small allotment of the sample before the results were finally compared, cross-checked, and written up together. The findings announced from that sample, published at the start of 2025, went further than anyone studying Ryugu or Murchison had managed before. Working under the direction of NASA chemists Daniel Glavin and Jason Dworkin, researchers identified 14 of the 20 amino acids that make up proteins in every living thing on this planet, alongside the same full set of 5 nucleobases already found in Ryugu, and a compound never before detected in any meteorite or return sample of any kind, tryptophan, one of the more complex amino acids used in human biology, appearing for the first time in material gathered directly from an asteroid. Tryptophan is a relatively large, complicated molecule, as amino acids go, one the human body cannot make on its own and must instead obtain entirely through diet, and finding a molecule of that complexity, surviving intact inside a piece of ancient asteroid rubble, suggested that the chemistry building these compounds out in the early solar system was capable of considerably more sophistication than the simpler molecules like glycine that had turned up in comets and meteorites up to that point. The Bennu sample also carried an unusually large quantity of ammonia, present in concentrations roughly 100 times higher than in ordinary Earth soil, along with phosphates, the phosphorus-bearing compounds that help hold genetic material together and give living cells their basic structural framework. Ammonia, in particular, caught researchers' attention because it is an unusually reactive ingredient, one capable of helping stitch simpler molecules together into the more complex amino acids and nucleobases found throughout the sample, less a finished building block itself than a kind of chemical glue that may have helped assemble several of the others. Bennu, like Ryugu, belongs to a class of especially dark, carbon-rich asteroids that scientists consider among the least altered bodies remaining anywhere in the solar system, essentially unchanged since the earliest days, described at the beginning of this story. which is precisely why both missions went to such extraordinary lengths and expense to visit them in the first place, rather than settling for one of the far more common, more thoroughly cooked asteroids scattered elsewhere across the solar system. When NASA announced the Bennu results at the start of 2025, the news traveled quickly, well beyond scientific circles, picked up by newspapers and evening broadcasts across the country. A rare moment when a rock gathered from deep space managed to compete for attention with the day's more ordinary headlines, if only for an afternoon. Even NASA's own announcement was careful to draw a firm line under the excitement, noting plainly that none of it amounted to evidence of life itself, only its raw, unassembled ingredients, a distinction the mission's own scientists repeated carefully more than once in the days that followed, and one that would matter a great deal to how the rest of this story unfolds from here. Researchers working across both missions, including the chemist Hiroshi Naraoka, who helped lead comparisons between the Ryugu and Bennu findings, soon noticed something else worth attention. Once the results from all four major samples, Murchison, Ryugu, Bennu, and a related French meteorite called Auguste, mentioned earlier in connection with an entirely different, less flattering chapter of its own history, were finally set down side by side. The balance between the two families of nucleobases, the larger purines and the smaller, more delicate pyrimidines, shifted consistently from one sample to the next, and that shift tracked closely with how much ammonia each sample happened to contain. Murchison, comparatively poor in ammonia, leaned toward the purine-heavy side of the ledger. Bennu and Auguste, both considerably richer in ammonia, leaned instead toward the pyrimidine-heavy side. Ryugu sat neatly in between the two extremes, carrying roughly equal amounts of both nucleobase families. A pattern this consistent, showing up across four unrelated bodies, studied by different teams using different instruments in different countries, suggested something deeper than simple coincidence. It pointed toward a single, shared chemical process, sensitive to the particular mix of raw ingredients available in a given corner of the early solar system, producing a slightly different balance of nucleobases depending on local conditions, the same underlying chemistry expressing itself a little differently from place to place, the way a single family recipe might taste subtly different depending on which pantry happened to supply the ingredients. No one sample, on its own, could have revealed a pattern like that. It only became visible once four completely separate bodies, gathered by separate missions decades apart and studied by separate laboratories on separate continents, were finally compared side by side. A reminder that patient accumulation, rather than any single dramatic discovery, is very often how a scientific picture actually comes into focus. This same primordial chemistry, it turns out, is not confined to our own solar system's asteroids and comets. In the summer of 2025, astronomers using a survey telescope in Chile spotted an object racing through the solar system on a path so extreme that it could only have arrived from interstellar space, from well beyond the Sun's own gravitational grip entirely. The third such interstellar visitor ever confirmed by astronomers anywhere. Unlike every comet native to our own solar system, bound in a long looping orbit that always eventually brings it back, an object like this one arrives once, sweeps past the Sun a single time, and then departs again forever, never to return, following a path so wide and so fast that the Sun's gravity can only bend it slightly rather than capture it. Later designated 3I Atlas, the object turned out to be extraordinarily old, its motion through the galaxy suggesting it may have been drifting alone between the stars for billions of years, quite possibly longer than our own solar system has existed, making it one of the most ancient objects ever examined directly by human instruments. As it swung past the Sun in the closing months of that year, telescopes detected water and carbon dioxide escaping steadily from its surface, along with an unusually high proportion of heavy water carrying that same heavier form of hydrogen mentioned earlier in this story, present in far greater abundance than in any comet native to our own solar system. Radio telescopes listened carefully for any signal that might suggest an artificial origin and heard nothing at all beyond the ordinary, natural hiss of an ancient icy body slowly warming in sunlight, settling that particular question quickly and firmly. 3i. Atlas has not, at least not yet, been examined closely enough to say for certain whether it carries the same amino acids and nucleobases already confirmed in Ryugu and Bennu. But its mere presence, an ancient traveler from another star system entirely, passing directly through our own on its way back out into the dark, confirmed at the very least that the raw material capable of building these molecules is not some rare, isolated feature of our own solar system's particular history. It is out there too, drifting quietly between the stars themselves, arriving here on its own schedule, entirely uninvited, in much the same way Murchison once had. Taken together, the 4 samples now sitting in laboratory vaults on 2 different continents, Murchison recovered from an Australian field. Ryugu and Bennu delivered home in sealed capsules built for exactly that purpose, told scientists something that decades of careful, patient, occasionally frustrating work had finally allowed them to say with real confidence. The molecular alphabet of life, the amino acids that build proteins, and the nucleobases that spell out genetic instructions, was not some rare accident confined to a single lucky rock. It was ordinary chemistry, repeated across asteroids and comets alike, embedded in the very material the solar system itself had been built from, and, if the fleeting glimpse offered by an interstellar visitor was any guide, quite possibly written into the raw material of other solar systems as well. 2 space agencies, working independently for the better part of a decade, spending together well over $2 billion, using entirely different engineering solutions to the same basic problem, had each come home with a version of the same answer, sitting today in vaults, barely a few hundred miles apart, on opposite sides of a single country. Neither mission had set out chasing a single dramatic headline. Each had been built, quietly and patiently, around the same modest, exacting hope that if you could only reach far enough, and hold still enough, and keep the sample clean enough, the rock itself would eventually tell you something no amount of clever guessing from Earth ever could. And yet, for all that this remarkable, patiently gathered inventory had settled, it left something else entirely untouched. Knowing precisely what these ancient bodies carry, down to the exact amino acid and the precise nucleobase, is a different matter altogether from knowing what became of that chemistry once it actually arrived somewhere, streaking down through a young planet's atmosphere in a burst of heat and friction, scattering across a scorched and violent landscape very much like the one described at the very beginning of this story. The vault in Japan and the vault in Texas, and the careful hands working inside each of them, could say with real confidence now exactly what these visitors were made of, down to the last stubborn nucleobase. What still remained an open question, and what would take a different kind of care altogether to answer honestly, was what any of it actually meant. Once the falling finally stopped, a sealed capsule in a nitrogen-filled glove box is a controlled, patient, forgiving place, in many ways the gentlest possible resting place a piece of the early solar system could hope to land in. The Earth, these molecules, would have actually needed to survive reaching billions of years ago, offered no such gentleness at all. Only heat, impact, water, and the crowded, ongoing business of a young planet already busy with its own violent chemistry. And that is a very different question from the one any vault, however well kept, can answer on its own. A sealed vault answers one kind of question beautifully and another kind of question not at all, and it is worth sitting with that difference for a moment before going any further, now that every meteorite, comet, and asteroid described in this story has finally been accounted for. Knowing exactly what a handful of asteroid dust contains, down to the precise nuclear base, tells you nothing on its own about whether that same chemistry could have survived the far rougher journey down to an actual planet's surface, through an actual atmosphere, into an actual ocean already busy with its own restless chemistry. That question turns out to have a partial answer already sitting quietly in plain sight, one this story touched on without pausing to notice it. Murchison itself, recovered from an open field in Australia, still carried its amino acids intact after a fall through the atmosphere bright enough to be seen for miles and hot enough to leave the outside of the rock scorched black. The reason those delicate molecules survived at all comes down to simple physics rather than any special luck. Stone is a poor conductor of heat, and the searing temperatures of atmospheric entry act mostly on the outer skin of a falling rock over a span of only a few seconds, leaving the interior barely warmed by comparison. Local residents who gathered Murchison's fragments in the hours after it fell described them as only pleasantly warm to the touch, never blazing hot, and that small, ordinary detail turns out to matter a great deal. A meteorite's charred crust is not a failure of the delivery system. It is the delivery system, an armor plate built entirely by accident, sacrificing itself layer by layer, so the cargo riding inside could arrive largely undisturbed. Engineers, designing the protective coating on any capsule meant to carry a human being safely home from orbit or from the moon, rely on exactly this same principle, built in deliberately rather than by accident. Let the outer layer absorb the punishment and keep whatever matters most shielded somewhere underneath it. Whether that same protection would hold up for a larger, more violent impact, the kind capable of leaving a crater rather than a scorch mark, is a separate and considerably less settled question. Some researchers have run experiments recreating the sudden, tremendous pressures of a genuine impact and found that the shock of collision itself can sometimes assemble simple organic molecules on the spot out of far simpler starting ingredients, adding a second possible source of raw material on top of whatever a comet or asteroid was already carrying when it arrived. Other researchers point out that the very same violence capable of building new molecules this way is just as capable of destroying whatever more delicate molecules were already present before the impact, and current evidence has not fully settled which effect wins out under any given set of conditions. Picture two cooks working the same stove at once, one of them assembling a fresh dish from raw ingredients thrown together by the heat itself, the other one accidentally burning whatever was already cooking on a neighboring burner, and you have a fair sense of the disagreement. Laboratory experiments recreating impact conditions have shown both effects happening side by side in the same brief instant, and untangling which one dominates during any single real impact on any single real planet has proven far harder than reproducing either effect on its own inside a controlled chamber. This remains an active and genuinely open area of research rather than a settled conclusion, and any honest account of this subject has to leave it there, unresolved, rather than pretend the matter has already been closed. What can be said with real confidence, gathered across every meteorite, comet, and asteroid sample examined so far, is considerably narrower than the boldest headlines written about this subject over the years might suggest, and it is worth stating plainly, one final time, exactly where that confidence begins and ends. Scientists have confirmed, through careful, repeated, independently verified measurement, that amino acids and the complete set of 5 nuclear bases used to write every genetic instruction in every living thing on this planet can be found embedded in ancient, unaltered material left over from the solar system's own formation, recovered from a meteorite that fell into an open field from the tails of 2 separate comets and from 2 asteroids delivered home in sealed capsules built for exactly that purpose. That is a remarkable, hard-won, thoroughly documented body of evidence. It is not, and was never, evidence that life itself crossed the emptiness between worlds. Every research team behind every one of these findings has said as much plainly more than once, precisely because the distinction matters enormously and gets lost with startling ease the moment a headline needs to fit onto a single line. That loss happens more often than it should. In the days after each of these announcements, versions of the same claim tend to circulate widely, stripped of their careful qualifications, that scientists have proven humanity came from space, that we are, in some literal sense, aliens ourselves. Nothing in any of the research described in this story supports a claim anywhere near that bold. A bold, simple claim travels faster and further than a careful, qualified one ever will, and a headline has no room at all for the difference between molecules and life. So the qualified version tends to arrive quietly. If it arrives at all, well, after the bold version has already been read, shared, and half forgotten. What has actually been found are ingredients, raw materials, the molecular equivalent of lumber and nails and window glass delivered to an empty building lot, not a finished house standing there with its lights already lit. A lot stocked with every board a blueprint calls for does not assemble itself into a house, no matter how complete or how sound that lumber happens to be. Something still has to do the actual work of measuring, cutting, and joining those materials in the right order, under the right conditions, patiently enough, and precisely enough to end up with something that can shelter, sustain, and eventually renew itself. Nothing found in Murchison, in Ryugu, or in Bennu answers that deeper question. It only tells us that whatever eventually raised the finished structure did not necessarily have to fell every tree and forge every nail itself, entirely alone, entirely on site. No chemist studying any of the samples described in this story would claim otherwise, and none of them have. The gap between a fully stocked building lot and an actual, standing house is not a small one. It represents, in living cells, an entire hierarchy of organization, molecules finding each other in the right concentration, arranging themselves into structures capable of holding a boundary against the outside world, and eventually finding some way to copy themselves faithfully enough, generation after generation, for evolution to have anything at all to work with. Delivering the lumber and the nails, however generously and however reliably, still leaves every bit of that harder work undone. It is worth pausing, before moving past that distinction entirely, to notice something almost as remarkable as the molecules themselves. Australian chemists in the 1970s, American engineers building aerogel collectors in the 2000s, Japanese researchers refining a gentler extraction technique decades later, and NASA scientists opening a sealed capsule in a Texas laboratory, all working in different decades, different languages, and under different national space programs, nonetheless insisted on the very same standard of proof before accepting any of this as real. The same handedness test, the same isotope check, the same refusal to call a finding confirmed until independent hands, using independent instruments, had reached the same conclusion twice over. That shared discipline, arguably, is as much a genuine part of this story as any single amino acid or nucleobase named within it. This distinction between delivering ingredients and delivering life itself separates the version of this idea now resting on genuine physical evidence from two much older, far more speculative cousins that share little more than a family name. The first of those cousins belongs to the very scientist who opened this story, the Swedish chemist who first proposed, more than a century ago, that living spores might drift between the stars on nothing more than the gentle push of starlight. That mechanism, as this story explained earlier, has not aged well under the scrutiny of modern instruments. Interstellar space bathes anything, crossing it in a relentless wash of radiation for a length of time measured in many thousands of years at the very least, an environment demonstrably hostile to the delicate molecules any living spore would depend on to survive the trip intact. Nothing found in any meteorite or any returned asteroid sample has done anything to rescue that particular version of the idea. If anything, the evidence gathered since has moved the whole conversation firmly away from it, replacing living travelers with something far smaller, far humbler, and considerably better suited to surviving such a journey in the first place. The second, stranger cousin belongs to a pair of scientists working more than half a century later, one of them among the most celebrated biologists of the entire 20th century, Francis Crick, who had already helped uncover the physical structure of the molecule carrying genetic information some 2 decades earlier, a discovery that had already earned him one of science's highest honors, and his colleague Leslie Orgel proposed, in a paper published in 1973, an idea considerably bolder than anything Arrhenius had suggested, that life on Earth might have been planted here on purpose, deliberately, by an advanced civilization from somewhere else entirely, perhaps as a calculated attempt to spread life, before that civilization's own world became uninhabitable. Crick and Orgel called this proposal directed panspermia, and to their credit, they framed it with real scientific caution, careful never to claim they had proven any such thing, only that the idea could not be entirely ruled out, given how little was then understood about life's earliest chemistry. Part of their reasoning rested on a curious detail of biology that still puzzles researchers today. Nearly every living thing on Earth depends on a rare metal called molybdenum for certain essential biological processes, an element that was never especially abundant or easy to come by on the early Earth's surface, which struck Crick and Orgel as an odd ingredient for life to have settled on if it had truly assembled itself here, from scratch, using only whatever happened to be locally convenient. Leslie Orgel himself, in later years, reportedly grew wary of how seriously some readers took the idea, gently reminding audiences that he and Crick had offered it as a possibility worth naming rather than a claim worth defending. The proposal never gathered any supporting evidence of its own in the decades since, and it could hardly be expected to. An idea built around the deliberate choices of some unknown, unreachable civilization offers no clear experiment capable of confirming or disproving it, no meteorite to examine, no capsule to bring home and study. It survives today mainly as a philosophical curiosity, a reminder of how strange the question of life's origin once seemed, even to two of the most careful scientific minds of their era, rather than as any kind of rival explanation to the evidence painstakingly gathered from actual rocks. History has been kinder to the modest, testable half of Crick's career than to this particular speculative aside, and rightly so, though it remains a genuinely useful reminder that even brilliant scientists, faced with a genuine mystery and an incomplete picture, will sometimes reach for an idea that later evidence quietly leaves behind. Every atom of carbon, nitrogen, and phosphorus sitting inside your own body tonight was forged, originally, inside a star that lived and died long before our sun ever ignited, one of countless furnaces scattered across an earlier generation of the galaxy, each one spending millions of years quietly building heavier elements out of simpler ones, before finally scattering that hard-won material outward, often in a single, catastrophic burst of light bright enough to briefly outshine every other star in its entire galaxy. That is a fact settled physics has never seriously disputed, confirmed independently by astronomers pointing telescopes at the glowing remnants of exactly these kinds of stellar deaths. This story has spent its time on something more specific and in its own way more remarkable still. Not simply that the raw elements came from dying stars, which has been understood for decades, but that at least some of those elements had already been arranged, out in the freezing dark between the planets, into the actual molecular letters of life's own alphabet, fully formed, before this world ever had the chance to write a single word of its own. That is not the same as saying you are, in any meaningful sense, an alien. And this story has no interest in pretending otherwise for the sake of a tidier ending. You were not assembled somewhere else and delivered here fully formed, the way one of Arrhenius's imagined spores might once have been. Nothing in Murchison, in Ryugu, or in Bennu suggests anything of the kind, and every scientist whose careful work filled this story would be the first to say so, plainly and without hesitation. But something quieter than that bolder claim, and in its own way sturdier, does seem to be true. Consider, for a moment, what is actually sitting inside you as you settle towards sleep tonight. The calcium built into your bones, the iron carried along in your blood, the nitrogen woven into every strand of genetic instruction inside nearly every one of your cells. None of that material assembled itself here, from nothing, the moment this planet happened to finish forming. Earlier chapters of that same story were already well underway, long before Earth existed at all, inside stellar furnaces far larger and far older than anything this solar system has ever produced. A part of the story so well settled by now that scientists rarely even pause to mention it anymore. This story has spent its time on a stranger, more specific chapter that comes after that one, a chapter only now coming fully into focus. Somewhere in the cold, unlit space between a young, still-forming sun and its outermost barely gathered planets, at least some of that same recycled stellar material appears to have been arranged, patiently and repeatedly, into the particular molecular letters. This story has spent so long tracing letters with names like uracil and glycine, sitting quietly in the dark, going nowhere in particular, waiting on nothing at all. They did not know, and could not have known, whether any world would ever come along capable of reading them. They simply existed, the way a sealed letter exists whether or not anyone ever opens the envelope, patient in a way only chemistry, with no calendar and no deadline of its own, can afford to be. Then, slowly, out of that same drifting cloud of gas and dust, a small, unremarkable planet began pulling itself together, cooling, gathering water, settling down from the violence of its own birth. And whether by one violent, glowing streak across a darkening sky at a time, or by countless smaller, gentler arrivals too faint to ever notice, At least some of those patiently waiting letters found their way down onto its surface. Whether that arrival made the difference between a living world and a barren one, no one can yet say with real certainty, and this story will not pretend otherwise, simply to reach for a tidier note than the evidence actually supports. Scientists are not finished asking these questions, and this story would be doing its own subject a disservice to suggest otherwise. Samples from Ryugu and Bennu remain carefully rationed out in laboratories on opposite sides of the Pacific, even now, with the majority of each sample deliberately held back in long-term storage for researchers not yet born, working with instruments not yet built, the same patient wager that once paid off so handsomely for Murchison decades after it fell. That wager has already paid off once before in exactly this field. The very fragments of Murchison examined by chemists in the early 1970s were nowhere near sensitive enough to find the delicate pyrimidenes that later instruments, decades on, finally teased out of that same rock, using nothing more than patience and cooler water. A spacecraft that once touched an asteroid named Bennu is already making its way toward another one entirely, a fresh chance to ask the same careful questions of a different, distant world. The same patient logic driving all of this work extends well beyond asteroids and comets alone. Robotic missions already being planned, or already underway, will drill beneath the frozen, cracked surface of Mars, in search of buried organic chemistry, sheltered from billions of years of harsh surface radiation. A NASA mission named Europa Clipper is already on its way to one such moon circling Jupiter, while a separate mission named Dragonfly, a small rotorcraft built to fly rather than roll across an alien landscape, is being prepared to explore the thick, hazy atmosphere of Saturn's largest moon later this decade, each of them designed in part to sample material flung out from hidden oceans that may have stayed liquid and largely undisturbed for longer than Earth's own oceans have existed. Farther out still, telescopes are only now becoming powerful enough to examine the thin atmospheres of planets circling other stars entirely, searching for the same basic chemical signatures this story has traced through meteorites and asteroid dust so far. Every one of these efforts is really asking a more patient version of the very same question that opened this story: whether the particular chemistry that eventually became a living cell on this one small planet is a rare, isolated accident or an ordinary, unremarkable outcome of chemistry that plays out quietly again and again wherever the right ingredients happen to gather in the right conditions. No single mission will likely answer that question on its own. It is far more likely to arrive the way nearly everything else in this story has arrived, gradually, sample by sample, laboratory by laboratory, patient measurement piling onto patient measurement, until one day, decades from now perhaps, the accumulated weight of evidence simply tips the answer one way or the other. And should any future mission ever manage to bring home a piece of a genuinely icy body, a comet nucleus, rather than merely its trailing gas, or a sample gathered from beneath the frozen crust of a distant moon, the same careful methods described throughout this story, the same handedness, the same isotopes, the same patient, unhurried chemistry stand ready and waiting to be applied there as well. None of this needs to keep you awake tonight turning it over, and it was never the intention of this story to send you off to sleep with a puzzle rattling around in your head rather than a settled feeling in your chest. It is enough, for now, simply to know that it is true, that somewhere in the quiet, ordinary chemistry, keeping you breathing steadily as you drift off, a little of it is older than the house settled around you, older than the sun that will rise outside its windows tomorrow morning, older, in the end, than the earth itself and that it has been patiently waiting in one form or another for longer than there has been anyone at all to wait for. Perhaps, somewhere on some entirely different world, orbiting some entirely different star, the very same quiet workshop delivered its own modest supply of ingredients long ago, and something there found a way, in its own time and its own manner, to put them to use, just as this small, restless planet eventually did. Perhaps not. Nothing gathered so far, in any vault, on any continent, answers that question either way, and this story will not pretend otherwise, simply to end on a more satisfying note than the evidence has actually earned. What can be said, plainly and without embellishment, is only this: long before this planet finished cooling, long before its oceans had fully gathered, and long before anything on its surface could rightly be called alive, the molecular alphabet capable of becoming part of a living thing was already being written, patiently and repeatedly, in the cold and the dark between the young sun and its outermost planets. It did not wait for an audience. It did not know an audience would ever come. It simply existed, the way the answer to a question can sit quietly correct for a very long time before anyone thinks to ask it. This small, blue, restlessly busy planet happened to be a place where enough of those patient letters eventually arrived, where enough time passed, and where something, somehow, in a manner still not fully understood, even now, began arranging them into a sentence of its own, a sentence that, all these billions of years later, is still quietly being written. Rest now, wanderer. The stardust that became you is in no hurry at all.